It's a real pleasure to have with us today Dr. Sobieszczyk, who is visiting us from New York City. She received her medical degree as well as her master's in public health from Columbia University in New York, and then completed a postdoctoral clinical fellowship in infectious diseases at Presbyterian Hospital of Columbia University Medical Center. She then completed a postdoctoral research fellowship at the Center for Infectious Diseases Epidemiologic Research at the Mailman School of Public Health at Columbia. She's currently an assistant professor of clinical medicine in the Division of Infectious Disease at Columbia University and is the director of their HIV vaccine clinical trials unit at Columbia's HIV Prevention and Treatment Research Division within infectious disease. And her research is focused on therapeutic strategies, integration of HIV prevention strategies, vaccines, microbicides, including addressing HIV infection in pediatric and maternal transmission in populations. So we welcome you and thanks very much for coming up to Albany today and sharing us about HIV vaccines.
Great. Thank you very much, and good morning, everyone, and good morning to our remote audiences. And I'm happy that my talk is marking the start of the summer season, which we badly need, I think. So what I'd like to do this morning is talk to you about HIV vaccines and some updates from the HIV vaccine research. But before I delve into some details, I wanted to go over some vaccine basics, because I don't think that many of you may be familiar with that topic. I'll touch upon some of the challenges with development of HIV vaccines, sort of that address the question of why we don't have a preventive HIV vaccine yet, and then talk to you about some key lessons that we have learned from recent and ongoing HIV vaccine efficacy trials that actually test whether or not a vaccine can prevent HIV infection. And then a brief kind of look into the future that will conclude my talk. So just let me start off just by kind of positioning the vaccines in the spectrum of the different opportunities for biomedical intervention that we have to intervene in sort of individuals' sort of potential exposure to HIV. So we know that there are sort of issues related to prior to exposure to HIV, and that's kind of where vaccines, preventive vaccines, come in. There have been some biomedical advances in the area of pre-exposure prophylaxis and antiretroviral therapy used as prevention. But I would venture sort of make an argument that a lot of these biomedical preventions really rely on the individual's behavior and on their adherence and compliance with those biomedical preventive interventions. And so the hope is then that a vaccine, a preventive vaccine, would circumvent some of those issues that have to do with adherence. So what types of HIV vaccines are currently being tested in terms of the design? So a lot of them contain either a sort of parts of the HIV protein or a peptide, or they have HIV genes or that are delivered through some sort of a vehicle that presents it to the immune system. And that vehicle oftentimes can be a DNA plasmid, and I'll show you some pictures of it, or a viral or bacterial vector that has spliced in it sort of inserts of the HIV gene. And the tricky thing is always to figure out not only what vehicle to use to introduce the HIV genes, but also which key genes to choose, which would induce a protective immune response. And then these vaccines are delivered in combination. So we sort of like to think about kind of these prime and boost strategies, that you would mix a DNA plasmid with some HIV inserts as a prime with a viral vector boost, or maybe a viral vector prime with a protein boost from the piece of the HIV envelope. So here's a couple of slides of sort of pictorial depictions of what I was talking about. So essentially, when you look at the HIV cartoon of an HIV virus, the inserts of interest that everybody wants to sort of include in an HIV vaccine in order to induce a protective immune response are the proteins on the viral envelope. And the thinking is that those are the ones that will induce an antibody, a protective antibody response, because that's the first thing that the immune system sees when it encounters the virus. Other HIV genes that have been included in the vaccines include the GAG, POL, and NEF sort of genes that are in the viral nucleus, and some of the core proteins like P24 have also been inserted. And so when you think about how those inserts are included in a vaccine, again, let me sort of mention to you that we're interested in finding vehicles or carriers that deliver and present these HIV inserts to the immune system. And those inserts, those vehicles can be a naked DNA plasmid, a bacterial or a viral vector that essentially you splice into them, the HIV genes, or the other option is that a vaccine would essentially sort of contain a synthetic HIV protein subunit, sort of similar to a hepatitis B vaccine. So how does an HIV vaccine work? Well, sort of the holy grail of a vaccine development, any vaccine development, is really to induce protective antibodies to neutralize the pathogen before it can enter into the cell, and thus prevent HIV infection. So that's definitely sort of one of the main goals of the HIV vaccine development. However, sort of we've recognized that the CD4 and CD8 responses are actually quite also critical in control of HIV replication, and thus an HIV vaccine will also aim to induce stimulate a productive CD4 and CD8 response, which will sort of act twofold, that they will both sort of augment antibody responses, but then also importantly, they will, especially the CD8 responses, they will destroy any infected cells. If, for example, a vaccine may not be able to prevent the virus from entering the cell, then the protective CD8 response may destroy the infected cell after the virus enters it and prevent replication. And thus, such a vaccine, sort of a T-cell inducing vaccine, would not only perhaps abort, but also control the infection and control the HIV replication, which has advantages not only to the individual, but also for public health. So again, just briefly to review, how might an HIV vaccine work? This is a slide that sort of depicts the trajectory of viral load following an exposure and infection. And there are several time points that we would want a vaccine to act. One is clearly before, sort of at the time or right before the exposure to HIV in order to prevent established infection. And this would be done by sort of having a vaccine that's already stimulated some pre-existing antibodies that are present and T cells that are present that have been induced by the vaccine that can stimulate this response. But if that is not possible, then other areas of opportunity are again sort of at the time of viremia, when the vaccine would sort of induce secondary T cell and binding or not neutralizing antibody responses in order to lower the viral load. So this is where the T cells come in, and the induction of the protective T cell responses is crucial. So what's been happening in the field to date? There have actually been over 30 vaccine products that have been tested since the HIV virus has been identified, and more than 85 trials that have been conducted, human trials from phase 1 to advanced phase studies. And there have actually been sort of thousands of volunteers that have invested their time and bodies to this effort, and it's been a multinational effort that's sort of been happening in the field. And yet, despite that, we clearly don't have a vaccine yet. So what have been some of the challenges in the vaccine development, and why don't we have a vaccine yet? And really, as I think you can all imagine, the HIV, so you all know, the HIV virus has presented a challenge not only to vaccine development, but to many other sort of areas of investigation in terms of even antiretroviral development of resistance. It's a sophisticated virus, one could say. And really one of the key issues are that there is a very narrow window of opportunity that we have to prevent establishment of infection. Because we know that right after exposure, there is a very early and massive depletion of CD4 memory cells in the gut mucosa. And the virus essentially establishes a reservoir pretty quickly, which sort of establishes, which is then difficult to eradicate. And so we really have a very narrow window of opportunity to intervene either with medications or with drugs to prevent these events from happening. HIV diversity is another challenge. As you can imagine, I'll show you some examples of that. There are multiple strains of HIV, so if we're trying to develop a vaccine against all these strains, As you can imagine, it presents quite a challenge. There is sort of the rapid replication and mutation rate of the HIV, which escapes from under the antibody or T-cell immune pressure, and thus you need to develop a vaccine that is sort of that is targeting that is inducing antibodies that are essentially immune to this viral escape, and then eliciting these or figuring out what antibodies can neutralize the virus has also been a huge challenge, and that really has to do with the envelope structure of the HIV itself. And so what I want to do is I want to just show you a couple of examples of what are the challenges related to this HIV diversity and to elicitation of neutralizing antibodies, because there have been some advances recently that have tried to circumvent these challenges that I think are very promising for the field. So the first challenge of the HIV diversity as to why we don't have a vaccine yet. As I mentioned, there are multiple strains of HIV. There's great sequence variability in the genome, which sort of leads to this very high antigenic diversity in the proteins that the immune system is trying to target. Essentially, it's trying to develop an immune response to a whole host of diverse antigens. And this slide depicts the global distribution of HIV subtypes and different recombinant forms. And as you can see, they vary across the globe. The predominant subtype here in the US is subtype B, but that is not the case in the rest of the world. So if you'd want to develop a vaccine, a universal vaccine, it would have to be sort of active against the subtype B and a C and D and other sort of, especially those that are most prevalent in the areas that need the vaccine, that are in the most dire need of the vaccine, such as sub-Saharan Africa. So the issue of the HIV variation, I like this slide because it depicts here the kind of the genetic variability of HIV, and genetic variable here, kind of this huge sort of dendrogram here, and the genetic variability of influenza A virus, both collected the same year in 1996. This is the HIV from Congo in 1996 and HIV influenza A virus from the same time period. And the size really of this tree depicts the extent of variation. So clearly, the HIV is much more sort of variable and diverse virus than influenza A. And if you think about it, we need to have a new vaccine for influenza every year, just because of the variability in the the antigenic shift and the drift. So if you think about it, we're trying to develop an HIV vaccine that's not only universal, but it sort of lasts for long periods of time and does not have to be remanufactured every year. So you can kind of imagine the conundrum. There have been some sort of advances, however, that have been promising in terms of how to circumvent this diversity of HIV, and that has to do with essentially formulating mosaics. So the goal is to increase the probability that the infecting strain of HIV that the individual comes in contact with would match what's in the vaccine, would match the vaccine antigen. But how can you predict that, right? Where the person gets infected, who they get the virus from. So what you're trying to do is you would develop these mosaics, so kind of different splices of antigens kind of contained into one vaccine that kind of optimize the cellular kind of immunologic coverage of HIV sequence virus diversity. So you're taking different splices of the genes and antigens from HIV viruses from all over the world, and you're introducing them in a mosaic fashion into a single insert into a viral vector on that DNA plasmid that I mentioned to you earlier. So that's I'm not going to show you that data, but there's actually been some promising results in non-human primate studies that have shown that such vaccines really induce immune responses against a whole host of viruses from around the globe, and those types of vaccines will be moving into human testing in not too distant future. So the other challenge that I mentioned has to do with inducing neutralizing antibody against the virus. Because that is really, if you think about it, if the immune system can have an antibody against an HIV that would bind to the CD4 binding site or some other area of vulnerability on the HIV virus, then it would prevent it from entering the cell and establishing infection. That has been a challenge just because of some inherent properties of the virus. But actually, recently, it's been recognized that there is that they're sort of been able to sort of really kind of narrow down the some of the key structures of the HIV virus itself, and sort of essentially define that the fact that it's the variable region of the HIV envelope that tend to be immunodominant, and they sort of induce kind of these neutralizing antibodies. And one of the other challenges has been that there's this glycoprotein shield around the HIV envelope that sort of makes it very difficult to have any protective interactions between a neutralizing antibody and those areas on the HIV viral envelope or viral epitope. So let me just show you a cartoon here as to what a vaccine is trying to do, essentially. So this is just a very cartoonish depiction of one of the spikes on the glycoprotein spikes on the HIV envelope. So a vaccine would ideally target some area of vulnerability on that HIV glycoprotein here, and would sort of prevent an antibody would bind to this area like a CD4 binding site and would prevent infection. What happens, however, in real life is that glycoprotein is a trimer. So there are actually 3 molecules of GP120 and 3 molecules of GP41 transmembrane protein that are visible to the immune system. So this, and what also happens is that area, sort of potential area of Achilles heel or vulnerability on the HIV virus, such as the CD4 binding site, for example, is actually not only variable, it varies from subtype B to subtype C, to sort of some of the other serotypes and clades of HIV, but also it's hidden by the trimeric molecule of the GP120. And not only that, but then there's this cloud of glycosylation that surrounds it, that makes it impossible for the antibody to bind to that sort of one Achilles heel that we know would sort of, an antibody to that we know would prevent the HIV from entering the cell. So what do we do? Well, there's actually been a lot of advances recently that have elucidated the fact that a small proportion, but about up to 25% of HIV-infected individuals, chronically infected individuals, actually have produced and have in them a neutralizing antibody. So they have an antibody which, when introduced into stacked up against a virus from a variety of individuals, would neutralize that virus. So it absolutely does no good to that already infected individuals because they're infected. They are just kind of, a manufacturing sort of these neutralizing antibodies. However, these antibodies have been isolated from cohorts in sub-Saharan Africa from infected donors, and they have allowed us to identify these key sites of vulnerability on the HIV envelope so that we know what those antibodies, what areas, what epitopes those antibodies are binding to, and those can serve as targets, sort of these kind of vulnerable sites on the HIV envelope that can serve as targets for vaccine development. So I'm going to show you some examples of these areas which we know these neutralizing antibodies from infected donors, they can bind to them. So it's CD4 binding site, no big surprise. But it's actually been elucidated exactly kind of, the structure of the antibody that binds to the CD4 binding site. And these antibodies have a variety of names depending on who's been developing them. There's the membrane, I'll show you sort of the membrane proximal region on the GP41 envelope. Some conserved regions on the V1, V2, and V3 loop on the GP120 envelope, which are sort of depicted here essentially. So these little red and green dots are essentially the binding sites of these neutralizing antibodies identified from the infected donors. And again, just to sort of orient you here, these are our glycoprotein spikes on the surface of the virus. We've got the 3 molecules of the GP120 and the 3 molecules of the GP41 transmembrane protein, and these kind of key areas of vulnerability that you're trying to elicit an immune response to. So what does that mean for vaccine? This is just another kind of depiction of the same trimeric structure, and I just wanted to draw your attention, I can find my pointer, to the VRC01 antibody that binds at or near the CD4 receptor binding site. That's pretty far along in terms of development and is going to be entering human testing this year. So as I mentioned, kind of the holy grail of vaccine development is really to design vaccines that induce the antibodies to these key sites of vulnerability on the HIV viral envelope. And there's VRC01, for example, and some others, but VRC01 in particular sort of entering human testing. One can also think about these antibodies not only as immunogens that can stimulate the immune system to produce such an antibody on its own, but they can potentially also be used as passive immunization or passive prophylaxis for HIV prevention, sort of akin to infusion of hepatitis B antibodies, sort of in an individual who's not immune and who had just gotten exposed. So those studies are also, those avenues are also being investigated. So I just want to mention one topic that has to do with this concept called HIV vaccine-induced seropositivity. Because if you pause for a moment and think about this, if we were trying to develop a vaccine that stimulates not only protective T-cell responses, but also these neutralizing antibodies, so some antibodies to the virus, you can think about that these antibodies, if in an individual who's receiving a vaccine that's being tested in clinical research, it can produce a false positive reaction on some HIV antibody-based tests. And that's sort of a phenomenon that has been well described. It really depends kind of on the vaccine components. So if there is a piece of the envelope gene present in the vaccine, then you can imagine that would stimulate an antibody and and also depends on the HIV assay that is used. And actually, the HIV Vaccine Trials Network has looked at about 900 or so blood samples from individuals who have been participating in vaccine trials, essentially who have received a vaccine that's been tested. And essentially, they've tried to determine whether they would come up positive on a Western blot or not. And what's been noted is here, I don't know if you can appreciate it, but in the table sort of depicts that really, again, depending on what was inserted in the vaccine, but up to 10% people who've gotten a vaccine that had an envelope gene have had a positive Western blot result, kind of on a Western blot test. And about 60% or so had an indeterminate test result. So this vaccine-induced seropositivity or seroreactivity is common, but it really depends on what's inserted in the vaccine. So what does that mean to the individual and to providers and clinicians who may be caring for them? That these individuals are HIV uninfected, but we always emphasize to them that it's critically important that they receive their HIV testing through the research site, where we do both an HIV viral test and we have a special HIV testing algorithm that sort of the lab, central lab, knows whether or not, sort of we're all blinded, but the lab sort of knows what kind of bands to expect because they know what kind of vaccine the individual may have received, and they compare it against their Western blot, and they can determine whether or not there is infection or not. We emphasize it to our participants because we don't want them to get tested outside, to get unblinded. Then they would know whether or not they got the vaccine and to sort of experience some potential social harm from being told, your Western blot, your HIV test is maybe positive or is indeterminate. We don't really know what's going on. You need to get further testing. So we actually, the Vaccine Trials Network and at our site, we have this sort of long-term follow-up protocol where we enroll all these individuals who've participated in vaccine studies, and we offer them free testing through the site. And not only that, but the protocol also sort of evaluates some of the newest platform for HIV testing to sort of establish whether or not the newer HIV tests, especially the combination antigen antibody tests, whether or not they still sort of produce these false positive reactions on the sera from individuals who've gotten vaccine. So let me switch gears a little bit and talk to you about some of the vaccine efficacy trials that have been conducted to date. And this slide is just a timeline that highlights that actually haven't been as many HIV vaccine trials. So there haven't been efficacy trials as there have been some of the phase 1 trials, because you can imagine it takes a while to move a vaccine product into efficacy testing. There have been, however, some studies that deserve mention and are important, like the VAX-03 or VAX-04 study that was called VaxGen. Some of you may have heard about it. It tested, it was sort of a monomeric subunit protein GP120, so it was sort of a protein vaccine that was tested both in the US and Thailand and in the Caribbean, and it absolutely had no efficacy because it was not, the vaccine was not able to elicit broadly reactive neutralizing antibodies. Then followed a study called the STEP study, which tested a viral vector, adenovirus 5 viral vector vaccine that had GAG-POL and NEF inserts in them, no envelope. And it was tested both in the North Americas and South America and in the South Africa. And it not only did the study vaccine didn't show any efficacy, but in a subgroup of individuals who had this prior immunity or had to adenovirus 5, and who were not, and men who were not circumcised, those individuals had a higher risk of HIV infection after receipt of the vaccine. And then the RV144, the Thai trial, which tested viral vector prime with an AIDS-Vax GP120 boost, and incidentally, this GP120 is the same GP120 that didn't have any protection in the earlier study. So this trial sort of was kind of offered not only a glimmer of hope, but kind of a boost to the field, because it was the first trial that had any modest, but any protective efficacy in humans in terms of reducing the risk of HIV infection. So it had very modest 31% reduction in HIV acquisition. And importantly, this study, unlike the earlier studies, had not only the presence of this GP120 protein given as a boost, but also the viral vector had an envelope from the HIV gene. So this is kind of the schema of this study. So it had sort of injections were given over a 6-month period. The pox virus contained the envelope gene, amongst others, and then the GP120 protein boost. And this is just summarizing some of the data, the Kaplan-Meier curves that kind of show the number of infections over time in the placebos versus the vaccinees, kind of demonstrating this, very modest but still protective efficacy of the vaccine. And what was notable about this study, that this protective efficacy was for very limited duration and really sort of peaked kind of at the 6 to 12-month period, and that's when the efficacy of the vaccine was highest when you looked at that snapshot of time, and it was as high as 60% actually, sort of it averaged to 30% over time. So there's actually a lot of investigation going on to sort of figure out how do you make that response, whatever immune response it was, how do you make it more durable than just beyond the first year? So what have we learned through this study? That it was a very modest, but still some level of protection. And there was a group of low-risk heterosexual ties. There was actually no effect of the vaccine on post-infection viremia or CD4 cell count. The efficacy sort of appeared to be happen early, and to be sort of short-lasting. And the vaccine also evoked not neutralizing antibodies, but something called binding antibodies against the virus. And I'll show you what I mean by that really very quickly decreased in concentration by about tenfold over 6 months. So they were short-lived. But this study, as you can imagine, kind of stimulated a lot of interest in trying to figure out what immune responses in those individuals who got the vaccine and who were protected, what immune responses were responsible for this kind of protective effect in determining whether or not a vaccine recipient became infected. And through a whole host, a lot of work, and lots of individuals who've investigated the sort of looking for these immune correlates of risk in this particular study, it was determined that there were sort of two specific measures, two main measures that determined whether or not a vaccinee had a higher or a lower risk of HIV infection. So one of them was sort of the levels of IgA antibodies that were binding to the envelope. That the higher they were, the higher level of those antibodies, the higher risk of HIV infection. And then conversely, the IgG antibodies to a V1, V2 loop on the envelope, the higher levels of that, the more protection against HIV in individuals who received the vaccine. So some of the hypotheses that were generated from this vaccine study, and again, is that individuals who had these high V1, V2 IgG antibodies, they received protection from vaccination, versus individuals who had low levels of these antibodies received less or no protection. And I'll show you in a second exactly what those IgG antibodies were targeting. And then conversely, individuals who had sort of low plasma IgA binding responses to the envelope were protected versus the high responses, high IgA responses received or generated less protection. So it sort of seemed like it was this yin and yang kind of a relationship that the IgG antibodies to V1, V2 may have contributed to protection from infection, and that was sort of mitigated by high levels of envelope-specific IgA antibodies. And here, what I'm showing you is again kind of our trimeric, sort of the GP1, the glycoprotein that's on the surface of the virion. And you've seen this before, kind of these three molecules of GP120. And I had shown to you earlier, sort of this is the V1, V2 kind of area, to which the IgG antibodies elicited by the Thai vaccine were protective. And interestingly enough, this is also the same area and same protective antibodies have been isolated from those chronically infected individuals that I mentioned to you earlier. So we know that the Thai vaccine was kind of zeroing in on that area of vulnerability of the virus, and it didn't induce a neutralizing antibody against that V1, V2 area or loop. But sort of an antibody that bound weakly, but still persistently enough, one could say, that it conferred some protection. So again, these findings are sort of being extended into other populations. There's sort of, I mentioned to you, there are attempts in sort of trying to induce a more lasting protective response, perhaps by giving another protein boost so that the production of these antibodies lasts kind of beyond the 6 to 12-month period. And those studies will be done in higher risk populations in sub-Saharan Africa and in Africa and in Asia to sort of see whether or not individuals who have higher risk of exposures and sort of higher viral sort of burden perhaps are also protected from a vaccine like that. So those are the three efficacy studies or several efficacy studies that I mentioned to you earlier. And at the same time, while especially the RV144 results were coming to the fore, there was another study, HVTN 505, another vaccine study that was about to get started. And I'll tell you a little bit about the HVTN 505 study because currently it's really the only efficacy study that was recently, the vaccinations were stopped and individuals are still in follow-up, but that's the only sort of recent vaccine efficacy study that we've been doing here in the U.S. So this particular study and the vaccine product was a DNA prime recombinant adenovirus 5 boost approach that was really originally kind of intended to stimulate these T-cell responses. The vaccine, one of the vaccine inserts was an envelope gene. So the hope was that it was an envelope gene from 3 different clades of the virus. So the hope was that it would stimulate the production of binding antibodies to the envelope. And it was also sort of focused on the key population that excluded individuals who had any prior exposure to adenovirus 5, because I mentioned to you briefly that the results of this STEP study sort of dictated that. This is the vaccine regimen. 3 injections of the DNA prime that amongst the GAG-POL and NEF inserts have an envelope gene in it, and then the recombinant adenovirus 5 injection that had GAG-POL genes and the envelope from the 3 different clades. And the objectives of the study were to really compare the number of HIV infections. We call them week 28 plus infections. So those are individuals who have completed all the vaccination. We are counting infections after completion of the vaccination series. And we also wanted to look at the viral load set point. So compare the average of the RNA levels in individuals who seroconverted on study to sort of see whether or not the vaccine had any effect on that. And the study enrolled 2,500 uninfected men who have sex with men who were fully circumcised, were serotype adenovirus 5 serotype negative because of the results of the STEP study, and fit certain behavioral risk criteria. And the study accrued over a period of 4 years. It started in 2009, which was shortly before the results of the Thai vaccine study came out, and it finished enrollment in March of 2013. And those are here the sites where the study was being conducted. It was only in U.S. We can talk about the reasons for why. And it was it was really sort of bi-coastal, and we had colleagues who conducted the study also in Chicago, Denver, and Colorado and Miami. I wanted to show you some of the kind of key landmarks about this vaccine efficacy study as it sort of evolved and as it enrolled over the 4-year time period from 2009 to 2013. Because as you can imagine, the HIV prevention field is not static, or even HIV treatment field is not static while a study is going on. So this particular study, in that study, we adapted to many of the events in the field, and thus the study sort of evolved with times. So actually, when it originally opened in 2009, it was much smaller than it was originally designed, and that was really just to sort of limit it to individuals who are circumcised, Ad5 seronegative, and to answer a very specific question about whether or not this vaccine can lower viral load, because the thinking was initially that it wouldn't be able to have protective efficacy. And then, as a result of the RV144 correlates analysis, which sort of showed that the presence of an envelope in the vaccine is critical because it stimulates production of antibodies. And as a result, and some of the non-human primate data that sort of showed that this particular vaccine was actually protective in non-human primates, the study size was increased to look at HIV acquisition as a primary endpoint. And not only that, but at the same time, the results of the PrEP study, IPREX, came out, which illustrated that daily Truvada had protective efficacy. And so we assumed that some of our participants would be interested in taking PrEP, and we sort of incorporated into design of the study some assumptions about PrEP uptake and what effect it would have on reducing the HIV incidence, and thus we would have to sort of increase the sample size to still see protective efficacy of the vaccine above and beyond any PrEP use. And so the vaccine study was increased. We also started collecting mucosal samples because we were interested in what the types of immune responses that are elicited in the blood, how do they correlate with what's happening in the mucosa? Because if you think about it, that in many individuals, that is the point of entry of the virus. So we may be stimulating all sorts of protective antibody responses in the plasma, but if they're not present to the same extent at the mucosa, they may do the individual no good. And then in 2014, actually, sort of about a month or so after we were fully enrolled, the Data Safety Monitoring Board sort of met and reviewed the unblinded data and determined that actually the futility criteria were met for both the HIV acquisition and HIV viral load endpoints, meaning that the vaccine was not protective and also didn't have any benefit on the viral load, and vaccinations were stopped. Participants were unblinded, were told about their vaccine assignment, and follow-up continues. We're following these individuals with HIV testing and referrals, et cetera. I want to spend just maybe a minute telling you a little bit kind of about some the sort of substantial community effort that sort of was mobilized to make this study happen. As I was mentioning to you earlier, as the prevention field was evolving, and so was kind of our approach to outreach and engagement of individuals. Social media and online advertising in 2009, when the study first started, were not utilized as a mode of engaging in doing community engagement, but they certainly were kind of about midway through the study when we realized that we would have a far better outreach if we used things such as Facebook, Twitter, and there were lots of kind of centrally coordinated campaigns that helped us disseminate the word about this vaccine study. And here's just an example of how, in advertisement, we monitored the kind of web traffic onto one of our websites, study websites, to kind of monitor the success of particular advertising campaigns. So if we posted an ad on our website, on some social media websites, we may kind of get some people who click through the ad and fill out a contact card. And then what we realize is that if you kind of refresh the ads, change the ads, clearly kind of stimulated a lot of interest, and we were able to capture that, and we were able to tailor our advertising or outreach based on the responses from particular ads. There were lots of very talented individuals who spent time doing this, and it was an incredible effort. And we also collected data kind of from every site to determine what strategies were most productive in terms of disseminating the word about the study and enrolling at the sites. So, and we also actually figured out that it was sort of direct person-to-person outreach and this internet and social media that had the most number of enrollees brought into the study. So let me show you some of the data from that, from the trial that was just recently published. So we enrolled a little bit over 2,500 individuals, but the analysis that I'm showing you here sort of is restricted to 2,496 participants. Median age was 29 years, so they were young. They were all supposed to be either male or transgender. We actually were able to enroll very few transgender individuals, but it's always a goal to make sure that everybody's included in preventive HIV prevention trials. We still had a predominantly white population. There was a lot of effort in trying to sort of have outreach to more African Americans and Latinos who are in greatest need of preventive modalities. And we've improved over what has been done before, but still have some way to go. And here's the data that sort of led to the decision of the Data Safety Monitoring Board to stop vaccination. So what I'm showing you here is the co-primary efficacy endpoint, which is HIV acquisition, week 28 plus infections. So infections in individuals who received the full vaccine schedule. It's a Kaplan-Meier curve. The black curve here are the placebo recipients, and the red curve are the vaccine recipients. And as you can see, there were slightly more infections in the vaccine recipients. This was not statistically significant, but importantly enough, there was no protective effect of the HIV vaccine on the course of the HIV acquisition. And I'm showing you some updated slides from an interim analysis that essentially confirmed that this is after we've unblinded individuals. We're following them, doing a frequent HIV testing. So this is data from this last fall, where we can see that actually the curves have flipped a little bit. There are just a handful more infections in the placebo recipients. Again, the differences in infections between vaccine and placebo are not statistically significant. The vaccine had no efficacy in terms of HIV, of protecting from HIV infection. And also, it didn't have any, I mentioned to you, it had absolutely no effect on the viral load among the individuals who got the placebo versus mean viral load of those who got the vaccine. One interesting thing that we're also following that's important is to make sure to sort of look at the study dropout. Are we losing more placebo or more vaccinees across the study? So this is data from before the unblinding, where there was a tiny, smaller rate of dropout in the placebo recipients. 6.6% of placebo recipients versus 4.8% of the vaccine recipients dropped out. This was just marginally statistically significant. It didn't affect our incidence, our HIV acquisition or results, because the difference was not significant enough. And it still is sort of below what we had assumed would happen in a clinical research study. We always assume we'd sort of lose about 10% of participants or so in the course of a research study, in the course of a follow-up. So this is still below that. Behavioral risk were sort of similar across the treatment arms, and I'm just kind of showing you here self-reported behavior of having more than 3 male partners or unprotected receptive anal sex. We collected a lot of behavioral data. And we also noted there was sort of the same between vaccine and placebo recipients, but we noted, of course, that those risk factors were predictive of HIV acquisition, not surprisingly. I'm going to show you just one slide, 1 or 2 slides kind of about the immunogenicity of the vaccine. So did the vaccine in a pilot study where we analyzed only a small proportion to date of uninfected vaccine recipients and placebos. We looked at whether or not the vaccine stimulated a cellular immune response and whether or not stimulated an antibody response, because as I mentioned to you, that's kind of the goal of a vaccine. So we actually noted that the vaccine was immunogenic, so it stimulated kind of, you look at these bar graphs, these red dots are immune responses elicited by a vaccine. And this is sort of placebo recipients who have no response as expected. And about, the vaccine sort of stimulated about 64% kind of cellular response to any antigen in terms of CD8 responses. So that's, pretty good. The other question was whether or not this vaccine stimulated any antibodies. And importantly, we noted that, yes, it stimulated production of binding antibodies. It did have the envelope gene, after all. But the one question that we wanted to ask is, how did those plasma antibodies compare to what was seen in the Thai trial, which had that modest protective effect? So how did those specifically plasma IgG and IgA responses compare? So if you remember, I mentioned to you that in RV144 was the IgG binding antibody to the V1, V2 region on the envelope of the virus that correlated with a decreased risk of infection. And what happened in HVTN 505, we actually noted that there was a lower response rate and magnitude of these vaccine-induced IgGs to that particular region. So it's sort of, we don't know what these binding antibodies mean, but what you're saying is in a modestly protective vaccine, there was a lot of these antibodies, and in a vaccine that didn't have any protective effect, those antibody responses were lower. So again, kind of, sort of confirming that stimulating antibodies to that particular region is possible with a vaccine and is important. And what about those IgA antibody levels, which in the Thai vaccine study correlated the higher levels? It sort of correlated with increased risk of infection. So actually, in 505, we had higher IgA binding antibodies to GP120, which were clearly not protective, and there was sort of a difference noted between the two regimens. So in summary, we noted that the 505 study was not efficacious. There was no evidence of any enhancement of HIV infection, and we're sort of following our participants and retaining them in the cohort. The humoral responses that were seen in RV144, that were compared to RV144, were present, but they were different from the RV144 vaccine. And those differences may actually be sort of understanding those differences in the antibody responses. I think we sort of all believe that are very important in terms of figuring out what are the correlates of HIV risk, what the vaccine needs to stimulate, and how to build on this in terms of designing new immunogens. So one could venture, I guess, sort of make a statement that even a non-protective vaccine can sort of lend a lot of information for the field and confirm or refute results of other vaccine studies. And I also just sort of want to, as we're closing off, just want to kind of pose the issue of designing and conducting these biomedical prevention studies in the era of pre-exposure prophylaxis. I think one of the questions that many of us are interested in is sort of evaluating how do the immune responses that are stimulated by a vaccine, how can they be augmented or perhaps not augmented by pre-exposure prophylaxis? So would administering pre-exposure prophylaxis in combination with a vaccine, with a partially vaccine, would that be synergistic, antagonistic, or additive? I think those are some of the questions that we're certainly interested in investigating and ought to be investigating. I think one of the challenges that will happen is we need to determine how to measure the vaccine vaccine efficacy results in the context of someone who is taking pre-exposure prophylaxis. As I mentioned to you, you can change the incidence in a small community that's participating in a vaccine study, and you won't know whether it's from a vaccine that's being tested or from the pre-exposure prophylaxis. I think a study that would aim to incorporate both or evaluate both would certainly be a more complex trial. It would sort of require a larger sample size. The good news, I think, would be that you wouldn't, if sort of in the future of the biomedical prevention, you may not necessarily need a vaccine that's 100 or even 95% effective. If you can combine it, you can have a partially effective vaccine combined with an antiretroviral that's either a short-acting or a long-acting antiretroviral that's also, taken intermittently, let's say, and partially effective. But if there can be a synergistic effect of the two, I think that's an added benefit to the individual. And I think really sort of presenting participants with more options of what modality to use will be key. So I think in conclusion, as we think about the HIV vaccines, the goal of a vaccine is to induce not only broadly protective antibodies, but also cellular immune responses that can control and prevent HIV infection, or prevent and control HIV infection. And I would sort of also pose to you that all the lessons that we're learning from small but also large efficacy vaccine trials can inform the design of future vaccines. And then as we're thinking about the different prevention technologies that are intersecting, I think this presents us with opportunities to evaluate them in combination, kind of achieve this incremental reduction in HIV incidence. The studies may become more complex, but I would also sort of say that more relevant to the individuals who are in need of these biomedical preventive options, and more applicable to diverse individuals who may want to use a vaccine or may not want to use a vaccine. They may want to use it in combination with condoms or microbicide or pre-exposure prophylaxis. I think investigating these options is incredibly important. So I would like to conclude by thanking you for your attention, but also acknowledging the immense effort that has sort of from lots of individuals, mainly study participants and the study teams that have designed and carried out these studies, and of course the funding agencies and developers of these vaccines. So thank you very much, and I'm happy to take questions.
We have a question from the internet. Can you hear me okay?
Yes, I can hear you.
Okay, the question is, I know AAA is not a vaccine, but what do you think of its use as a treatment for HIV?
So the question was about AAA as a treatment for HIV, and it's not a vaccine. I think it's a drug that has actually, when you kind of think about the history of HIV treatment, there have been many landmarks. One of them has been the development of protease inhibitors, which in the mid-90s have really impacted the mortality and morbidity of individuals living with HIV. The regimens were still complex and difficult to take. And then with the advent of a drug like a triplet, which is a combination of three agents, that has also revolutionized HIV treatment for many individuals who are, had sort of in terms of mitigating the issues of drug burden and number of pills. So in that respect, I think it's been a great treatment option for many individuals. These treatment options, I sort of feel, are always very individualized, and decisions have to be made between the patient and the providers, and many things have to be taken into consideration in terms of whether or not it is the right treatment for them. Thank you.
Two questions. One is, can you took into account in this 505 study the pre-exposure prophylaxis, but was it actually measured? part of your question there. And then the second question I have is, do you think the negative results of the vaccine trials are going to significantly impact recruitment for future vaccine trials?
Yeah.
Could you repeat the question, please?
Yes, I'll repeat the question. So one was, I mentioned that we incorporate in the 505 study, we incorporated assumptions about pre-exposure prophylaxis use, and we educated participants about it, et cetera. But did we actually measure it? And to answer your question is yes, we did. We've had questionnaires that have collected, self-reported, they ask individuals to self-report whether or not they were taking PrEP or post-exposure prophylaxis and how and when. Some of those questionnaires were administered by the study staff and others were done by a CASI, so the individual was just answering the computer. So you can sort of measure the differences. And then we also collected and stored blood to measure the drug levels, actually, to sort of correlate that. I didn't show you some of the results about the PrEP and PEP uptake, which actually was not very high at all in our study, but we don't have all of the data analyzed yet. And then your second question was about, would results of a disappointing or a negative or, a vaccine study that's not efficacious, does that negatively impact the efforts to recruit into upcoming vaccine studies? And I actually think, it presents a challenge to researchers and staff in making sure that we are able to explain the results of a study very clearly, not only to our participants, but to our community members. And also emphasize the fact that even though a vaccine may not have been efficacious, we're still learning a lot from it, and there's sort of a whole, suite of studies that are sort of being done and generated as a result of this vaccine study. So I think couched in those terms and kind of emphasizing that vaccine development is an iterative process. So you may have a series of weekly positive, negative, promising, not so promising results, but they all feed, just like in clinical research with HIV treatment, they all feed into a body of knowledge about HIV vaccines that we're using to sort of design next immunogens. So I think explaining that and being very transparent about it and making sure that we sustain the effort in terms of community engagement and explaining of the results and keeping this issue at the forefront of people's agenda, I think it will. I don't think that it will negatively impact future efforts. And it's actually a lot of, as it was showing you the community engagement mobilization efforts. I think a lot of relationships have been forged over time that will be beneficial for other studies. More questions? Yes.
Hazard one. Comparing your last study, the RB-144, I see two differences in the populations. One had to do with being mainly MSMs versus heterosexuals, which means different routes and different first tissue of exposure.
Correct.
And the second is because a likely difference in the primary subtypes that these groups were exposed to. What lessons might that have for the additional difficulties of
Yeah.
preparing
Yeah.
vaccines?
So thank you for that question. So let me just repeat it quickly. So you were mentioning that one of the key differences between the HVTN 505 study done in the U.S. versus the Thai study were populations, MSM versus in U.S. versus heterosexual low-risk individuals in Thailand. And then the different subtypes of HIV that circulate in those regions, and what are the lessons that we're learning from these studies. Does it present any further challenges for vaccine development? So that's a great question, and I think that's one of the. You're absolutely right, because the MSM population, different route of entry of HIV, different also the amount of the incidence in that population, HIV incidence was also different, and so the rates of exposure or encountering the virus were much higher in the MSM US population in our study than in Thailand. So one could sort of ask, how would the Thai vaccine stack up against in a higher risk population where there is especially in MSMs, where the portal of entry is different and the viral exposure is also different. So that's why that vaccine is actually being redesigned to be translated into a population that has a different subtype of HIV, so into sub-Saharan Africa, where subtype C is the predominant one. So the vaccine inserts will be changed, and that takes some effort, and it will be measured for efficacy in that higher risk population to see whether or not it can still have the same protective effect, modestly, but protective effect. And we don't know. It needs to be tested. But that's sort of one important question, because the subtype issue, the Thai vaccine study was manufactured specifically for the subtype that's predominant in Thailand. So it has to be reconfigured and remanufactured to test in sub-Saharan Africa. But I think some of the lessons that are being learned from the RV144 study is that induction of these antibodies, even in a low-risk population, was modestly protective. And we're seeing kind of the converse finding in 505. So I think, if anything, it's kind of lending more credence to the fact that these, if you can stimulate these binding antibodies in another population, the vaccine may also be, or similar antibodies, the vaccine like that may be modestly protective. So we're just sort of building on those results, but the vaccine has to be reconfigured into another population.
I just have a question about the antibody breadth for the RV144 and the HVTN 505. So in the RV144, the antibodies to the V1, V2, they cross-reacted with lots of subtypes, V1, V2. Has that analysis been done for the 505?
Not yet. Yeah. It's a good question.
You want to repeat your question?
Oh, sorry. We're just talking about for RV144. We're sort of mentioned that it's the the antibodies to that V1, V2 loop that were sort of associated with some modest protection. And have we looked at the different subtypes of antibodies that were stimulated in 505? So some of that work has been done, and it will be presented sort of shortly, but hasn't been finalized yet. Yeah. Thank you.
Well, thank you very much, Dr. Sobieszczyk. That was really concise and superb a review, and good for us clinicians to kind of hear the whole story. So thank you very much. I'd also like to thank our funders, the New York State's Clinical Education Initiative and the New York, New Jersey AIDS Education and Training Center. Thanks all of you for coming today, and hope to see you at our June symposium, June 3rd and 4th at the Hilton Garden Inn. Thanks very much. Have a good day.